Welcome you to BYJU'S 9th and 10th grade channel. I'm your biology teacher Aishwarya, and in this particular video, we are going to be looking at all the activities which are there from the chapter Life Processes. Now, of course, on a board examination front, this video is going to be very important for all of you because you get short 1 mark or 2 mark questions in your exams. Now, they may ask you direct questions or they can ask you inference-based questions. So, understanding these activities across science chapters are important for building your competency. So, I request that before you get started with this video, please have your textbooks and pencils ready with you so that as and when I tell you the important pointers, you can mark it down in your textbook. And of course, this video is going to be your one-stop solution in understanding all the activities from life processes. So without wasting any more time, let's get started. So the first activity that we have here is activity 6.1, which is there in page number 96. And this particular activity is related to the concept of photosynthesis. So let's go through it step by step. Now they're saying that we have to take a potted plant with variegated leaves. For example, money plant or crotons. So here when we say that is a variegated leaf, it means that it is a leaf. The example that is given right here, wherein we see that there are some green parts, right? And we see that there are some non-green parts. Yes? So that is what we simply understand as a variegated leaf. There are some portions of the leaf which have green in color, while some that are not. Yes? Now we are saying that we will take this plant. Okay, so imagine I have a potted plant with me, right? And I take this particular potted plant with the variegated leaf and I keep it in the dark for 3 days, right? And they're saying that we have to keep it in the dark for 3 days so that all the starch gets used up. Now, when in the dark, you know that plant is not going to photosynthesize. That means that they're going to depend on their reserve energy, which is why this process is what we call, maybe I'll use another color for better understanding, right? This step that you see right here that I am describing is what we call as de-starching, removing all the starch, right? Now from here, after we keep it in the dark for 3 days, we are now going to take it out and then keep it in sunlight, right? So we're going to keep it in sunlight for about 6 hours. Now we're saying that we have to take 1 leaf, okay? Experiment is happening only with 1 leaf. You take 1 leaf and you mark all the green areas and then you trace them onto a sheet of paper. Okay, so we need to know what are all the green parts, right? So we trace it out and we keep it handy for us later. Then what are we doing? Then comes the interesting part. Yes? So I have my leaf with me. So maybe I'll use a green color. Now I have a variegated leaf with me. And we're saying that we're going to take this leaf and we are going to first boil it in water for a few minutes, right? So I'm going to take this and I am going to put it in boiling water for a few minutes. Now, why are we putting it in boiling water? Because when you boil it in water, we see that the cells of the leaf will rupture, right? So we want to test for the process. We're looking for something very specific. So we need the cells to rupture, which is why we will dip in the leaf in boiling water for a few minutes. Now, followed by this, we are going to take the same leaf, okay? So, we're going to take this leaf and then we are going to put it onto a beaker which consists of alcohol, right? And here we're going to say, or what we're going to do is we're going to immerse it in a beaker containing alcohol. Now, we will take this particular beaker and immerse it in a water bath and heat it. So, we will not boil the alcohol directly, but we will place this into a water bath, yes? And then we will boil the water. So this is what we call as a double water bath, right? So we're not directly boiling the alcohol, but we're boiling the water around it, which in turn is going to boil your alcohol, right? And your leaf is also kept inside this, right? Now your leaf is also inside. Now why are we boiling it in alcohol? We boiled it in water to rupture the cell. Now we boil it in alcohol to remove the chlorophyll. So this step right here is to remove the chlorophyll that is present there. So that no more photosynthesis takes place, right? We want to do a proper test to make sure and we test for starch that is there, right? So we are ensuring that we remove all the chlorophyll. Now, what is going to happen to the color of the leaf? And what is the color of the solution? Now, we have removed chlorophyll. That means that the color of the solution that is there is going to be green, right? So here, color of solution is going to be green. And what will happen to the leaf? The leaf is going to lose its color because we have boiled it in alcohol. Now dip the leaf in dilute iodine solution for some time. So now that we have done all of this, what are we going to do? Right? So I'm going to take a white color itself. And now here we are going to be adding some drops, right, of iodine solution. Now at this point, we see that we add a few drops of iodine solution and we take out and we rinse the leaf, right? So we've added iodine. Now we will rinse it off. What is the observation? Yes. Now going back to the previous page, right? You see that the result is given. This is the variegated leaf before and this is what you get after. Now notice how the parts, right? So quickly going here. Notice how the parts wherever it was green has now turned dark blue in color, right? And of course, basing our knowledge on starch test, right? So if I have to write the starch test for all of you, we know that when we add iodine to starch, right? So when you add iodine solution, which is normally like a brownish color solution, if starch is present, right? So if starch is present, then you know that the complex that it's going to form is going to be blue-black, right? So if blue-black is there, that means that starch is present. So in the green parts, wherever you see dark blue, starch will only be present if photosynthesis has taken place, right? We know that end product of photosynthesis is glucose. Excess of glucose is stored as starch. So wherever the green parts are there, you see that starch test turns positive. That means that for photosynthesis to take place, chlorophyll is essential. What is our inference from this? What do we understand? We understand that wherever the regions where chlorophyll was present, we got positive for starch test. That means photosynthesis will happen only if chlorophyll is present. If chlorophyll is not there, you see that it will be negative for starch test. That means that photosynthesis has not happened. Easy peasy. So you need to break down these activities step by step in order to understand better. So quickly, let's move on to the next activity, which is activity 6.2. Now talking about activity 6.2, it is present in page 97 itself. And this right here is a very simple activity. Now here they've also given you a diagram. Now understand that this right here can come for you as a diagram based question also. So everybody please put a star mark under figure 6.4 because this can come for you as a diagram based question as well in your boards. Yes. So let's have a look at this particular activity. Now take two potted healthy plants. So we've taken two potted healthy plants which are nearly the same size. Preferably take two same plants, right? And of course keep them in a dark room for 3 days. Now very quickly pause this video and try to answer this. Why are we keeping them in the dark for 3 days? We know the answer. For the process of destarching, to remove the starch out of it so that we can test for photosynthesis. Now what we're going to do is we are going to place each plant on a separate glass plate. Okay. So put them on a glass plate and place a watch glass containing potassium hydroxide. Students, very, very important pointer here. We are taking a watch glass containing potassium hydroxide or KOH. Please star mark this, right? And we see that it is there on one side. Now in your experiment only, they have told you what potassium hydroxide is doing. Potassium hydroxide is used to absorb carbon dioxide. Easy experiment. If potassium hydroxide is there, it is going to absorb all of your carbon dioxide. Now I have kept potassium hydroxide in potted plant A, while potted plant B has no potassium hydroxide. So now I have left it, I have covered both with bell jars. Both are covered with a bell jar so that you know it is protected. And I have left it as it is for some time. And just to make sure that there is no entry and exit of air, right, from any of the corners, I have sealed it with Vaseline also, so that it doesn't disturb my experiment. See, covering it with a bell jar, sealing it with Vaseline, it's only there so that we make sure that our experiment happens properly and there is no interference with our experiment, right? Now we leave it in sunlight. So let me remove some of the annotations. So now what am I going to do? Now I am going to leave it in sunlight for 2 hours. Yes. So I'm leaving it in sunlight for 2 hours. Photosynthesis is going to happen. Then what do? I will take one leaf, right? So I'm going to take leaf from plant A. I'm going to call that as leaf A. And then I'm going to take leaf from plant B. So I'm going to call it as leaf B. Yes. So let's visualize this a little bit. All right. So now I have 2 leaves with me. No, I have leaf A. Yes, what did my leaf A? It came from the potted plant which had KOH, right? So I'm going to put a tick near KOH. While leaf B that is there came from leaf plant B which had no KOH. Now we left it for 2 hours and now what are we going to do? We are going to do the same starch test. So we're not describing the steps. Put it in boiling water, then put it, in alcohol, then, add iodine solution. Now, which one will turn positive for starch test? Is it going to be leaf A or leaf B? Pause this video and try to give the answer yourself, right? Try that first and then you can watch ahead with the remaining explanation. So now we know that leaf A was in a bell jar that had potassium hydroxide that absorbed all my carbon dioxide. Carbon dioxide is necessary for photosynthesis. It is a raw material for photosynthesis. So has photosynthesis occurred in this particular leaf? No, because raw material was not available. But what about leaf B? There was no potassium hydroxide. Nothing hampering the process. Which means in this particular case, photosynthesis has happened. So when I add my iodine solution to this, what is going to happen? You evidently see that or when I test for starch, you see that it will turn positive for starch test because photosynthesis took place. So from this, what can we infer, right? What do we understand? We can infer or we can observe that carbon dioxide is necessary for photosynthesis. It's one thing for us to just learn that carbon dioxide is necessary, but by carrying out this experiment, you realize that if CO2 is not there, it is not going to take place. So I hope you have made a note of all the important pointers that I have told you about in this particular experiment. Moving on, of course, if you want to know more about photosynthesis and experiments related to it, because these were 2 that are mentioned in your textbook currently, but there are more such experiments. And if you want to know more, there is this amazing video which has already been done by Ankita ma'am on the channel. The link will be available in the description of this video, and you just need to go check it out, where she has explained everything in great detail, even learning about aquatic plants and how various other factors are important. Through experiments, we will learn about how they are important for photosynthesis. So very quickly, students, let's move on to activity 6.3, right? So everybody requesting you to move to activity 6.3, which is there in page 99. And this particular activity is related to digestion. So let's go at it step by step. Yes. So as you can see, we are taking 1 ml of starch solution in 2 test tubes. So let's visualize this, right? Makes things easy for us. So let me quickly go ahead and draw 2 test tubes, okay? So I have test tube A with me. They have asked me to take 1 test tube here. And of course, we have test tube B. Now, of course, it's at a little bit of the bottom of the screen because I'm not very tall. So let's make do with it, right? So we have 2 test tubes A and B. And we have to take 1 ml of starch solution in it, right? So we are taking, so let's assume I have put 1 ml, right? I have added what? 1 ml of 1% starch solution in both the cases, okay? Now, of course, what I'm going to do is I'm going to add. So let me go to the other side. So now, of course, I'm going to add 1 ml of saliva to test tube A. So to test tube A, I am going to add what? 1 ml of saliva. Now, am I adding anything to solution B? No. Solution B is going to be left undisturbed. Yes. And now what we're going to do is we are going to leave this as it is for 20 to 30 minutes. Yes. Now both of it. So here, right here, what do you see? Only starch is there, right? So this has only starch solution. Yes. So now after this, what we're going to do is we're going to add a few drops of iodine. Now, of course, I will use a dark blue color. We're going to add a few drops of iodine to both, right? So these are few drops of iodine solution. Now, what is the question asking you? The question is asking you, in which test tube do you observe a color change? Is it going to be test tube A or is it going to be test tube B? Pause this video and try to answer this question by yourself. You know what is given to you. Try to answer it. Where are we going to observe color change? Now we know, right? We know that we will observe color change in test tube B. Yes? Now the next question is asking us, what does this indicate about the presence or absence of starch in both the test tubes? So we need to identify if starch is present or absent. Now I can pause this video and try to answer it yourself, because now you've got a hang of it, right? You've got a hang of it. So now, of course, when I talk about A, is starch present in test tube A? It is not, which is why I did not see a color change in my solution. Is starch present in test tube B? Yes, which is why I observed a color change. Now from this, it goes to a very important question, right? What does it tell us about the action of saliva on starch? Very, very important. So now, of course, when we talk about saliva, right, we know that saliva that is there is a watery fluid which has an important enzyme which we call as salivary amylase, right? So we know that when starch that is present in our food is, when we ingest the food in our mouth itself, starch is broken down into simpler sugars. Yes. So we know that it is broken into simpler sugars like maltose in the presence of salivary amylase. Yes or no? We are all aware about this particular reaction. So in test tube A, what did I do? In my test tube A, my A not only had starch, but it had saliva. So because it had both starch and saliva, this particular reaction that I have written has happened, which is why is starch present? No, starch is not present. We have simpler sugars, which means if I add iodine, am I going to get a result? Absolutely not. But in the case of my test tube B, it has only starch solution. So it will turn positive for the iodine test or the starch test, right? So with this, of course, what can we say on the action of saliva on starch? It breaks down. Saliva acts on starch and digests it and breaks it down into simpler sugars. So now I hope that this particular activity has become easy. This activity has a high percentage chance because in the previous year board exams that happened in 2022-2023, this question was a very popularly asked question. So please put a star mark. And whenever you get such questions, draw and represent for easier understanding. Now let's move on to activity 6.4. Now this is a very easy activity, right? So now let's have a look at it. Let's take some freshly prepared lime water in a test tube. Okay, so let's get to our drawing board, and as you can see, I have taken my test tube. Yes, and what have I put? I have put lime water, right? Now lime water, and now of course, blow air through this lime water, right? Now note how long it takes for the lime water to turn milky. Okay, so now what we're going to do, we are going to take a straw, right? And then we are going to blow, right? So from there, if we blow, we are saying that this lime water is going to turn milky, or it will turn a little more whitish in color, right? Like hazy in color. Now this is going to be one thing. Now the next one is you take another test tube. Okay, let's go to our drawing board again. So you have two test tubes with us at this point. So test tube 1, test tube 2, let's call this A and let's call this B, right? So in this I have taken lime water, right? So in this I have lime water, then I blew, and then it turned milky. Yes. Now what are we going to do in test tube B? I have taken lime water, right? Then what I'm going to do is I will take a pitchkari, right? Pitchkari, or take an injection, whatever it may be. But I'm just going to draw, right? Just representing it simply. And we're taking a pitchkari, which will take all the natural air in the surroundings, right? And then we are going to inject it. And then they're asking us, see how long it takes for this lime water to turn milky. Now the time that is taken in A is different from the time that is taken in B. A turns lime water, the lime water in A turns milky slightly faster than in B. Now, why does this happen? Actually, for B, it will take quite some time before it turns milky. Now, the question that they are asking us is, what does this tell us about the amount of carbon dioxide that we breathe out? Now, when we talk about amount of carbon dioxide, in the case of the pitchkari, it's taking air from the surrounding, right? Which means that in our surrounding, there is 0.04% of carbon dioxide. Yes, percentage is considerably less. But when we blow, that's the exhaled air, right? The air that we blow out is the exhaled air, which has 100 times more carbon dioxide, roughly 4% carbon dioxide, because the exhaled air has all the carbon dioxide that is produced as the end result of respiration, which is why when carbon dioxide reacts with lime water, it turns milky. So now I hope that the answer to this particular question or this particular activity has been easy. This activity, of course, is there in page number 101. So I hope you've made a note of this particular activity. Now let's move on to the next one, which is there in the same page, 101. But this is a very easy one. Yes? So take some fruit juice or sugar solution and then let's add some yeast to it. Okay? Focus on this part. Let's add some yeast to this. Now take this mixture in a test tube and have a test tube fitted with one hold cork. Alright, so this means that, let me just move on. Okay, so now I have taken a test tube. Yes, so let's draw a test tube. Most of our activities are related to test tube. So now I have added what all? I have added sugar or maybe some natural fruit source and I have added yeast, right? Now I have tightly sealed it with a cork. Okay, I have tightly sealed it with a cork. So I'm just going to make sure that it is tightly sealed. Now I have made sure that I have only, oops, just give me 1 minute. Yes, so I have made sure that I have opening only for one straw, like a U-tube, like a tube kind of thing, right? It's a bent glass tube, right? So it is bent and it goes into another solution which has lime water. Now you know where this experiment is going to. At this point, you don't even need me to tell you, you know the answer. But nonetheless, right? So dip the free end of the glass tube into a test tube or a structure containing freshly prepared lime water. What change is observed in the lime water? Okay, first thing, what change is observed in the lime water? Students, pause this video right now and try to answer it yourself. You have sugar and yeast on one end attached to lime water, right? And they're asking what change is observed. So to directly give the answer, we know that the lime water that is there will turn milky, right? So the lime water here is going to turn milky. Second part of the question, how long does it take for this particular change to happen? Now this is going to take some amount of time, right? It's not like it's going to be immediate, but we see that it takes a little bit amount of time for this change to happen. What does this tell us about the products of fermentation? That is the third question. What does it tell us? We know that in fermentation, sugar is broken down, right? In the temporary absence, right? Because we know that this is tightly fitted, but still breakdown of sugar is happening by yeast, wherein we see that alcohol is produced. Yes. So we know that alcohol is produced and as a byproduct, carbon dioxide is given out. And this carbon dioxide is given out as a gas, which of course travels up, reaches your lime water, reacts with lime water and makes it milky. Yes? So now we know the answer to all the three questions. Now this is a pretty straightforward question. Now the next two activities which are there, activities 6.7 and 6.8, are a little bit of data-driven activities, and this right here, mostly so far from what we have observed, they may or may not come, right? They are not very, very important up until activity 6.5, which is also pretty important because they can ask you. But 6.6 and 6.7 are pretty easy ones, but does not mean that we take it lightly. We go through it nonetheless. Observe a fish in an aquarium. They open and close their mouths and the gill slits or the operculum which covers the gill slits behind their eyes also open and close. Are the timings of the opening and closing of mouth and gill slits coordinated in some manner? Now this of course is a yes or no answer. Now at this point of course we see that the answer is yes, because we know that the oxygen that is there is dissolved in the water, it enters through the mouth and then of course it reaches the gill slits, right? So we see that at this point it is coordinated. Now, of course, here is then an observation-based one where they're asking us to compare the breathing rates, which is a data-driven thing, right? So you can find this out, but again, they'll not ask you to count the number of times the fish opens and closes its mouth in your board exams, right? You don't have an aquarium right in front of you. They may give you this data, they may give you the number of times we breathe, and then they may ask you to identify what is the difference in breathing rate in a human and breathing rate in a fish, right? And we know that breathing rate in fish is higher because of the dissolved oxygen, right? And we know that that is very, very little, which is why the breathing rate is higher, to make sure that more oxygen is made available. So the second set of questions will be given to you in this format, and they'll not ask you to sit and count how many times a fish is doing, or they'll not ask you to count how many times you are breathing. But this part of the question can be twisted and asked you in terms of breathing rate. So you can put a star mark and write the part that I've just told you about. Now let's move on to question number 6.7, activity 6.7, which is there in page number 105. Now this is also a little bit of observation driven one. So either they will give it to you and they'll ask you to, sort of identify what is the observation. But again, chances of such kind of questions where, they ask you to observe will be simple. But like I said, they will give it to you and then you have to identify. So the question here is, visit a healthcare center in your locality and find out what is the normal range of hemoglobin in human beings. Is it the same for children and adults? Now, of course, we see that in children, of course, the amount of hemoglobin can vary. The percentage might be between 9.5 to maybe around, 12 or 13. Now, of course, in adults, it will be slightly more between 14 to 16, right? So that is the amount that they calculate. Is there any difference in the amount of hemoglobin levels for men and women? Yes, of course, we see that there is a slight difference in the range. We see that men tend to have slightly more than women, right? Of course, the number varies. Then, of course, here they are asking you to visit a veterinary clinic in your locality and find out the normal range of hemoglobin in an animal like buffalo or cow. Again, this is not something that they will ask you to, find out at that given point. This is something that they might give it to you, right? They may give you the hemoglobin level in a cow. They may give it to you how much is there in male and how much is there in female, and they make you maybe identify what is the difference. So the question could be, given to you in that kind of a manner. But again, there are only slight differences that we observe in the range of hemoglobin. And we know that hemoglobin at the end of the day is extremely essential in living organisms. I mean, especially in mammals, right? Because we know that they help in transport of oxygen. So with this, of course, everybody, activity 6.7 is done. Like I said, this particular activity is very observation-based driven, but they will normally give it to you and we will be solving such kind of questions wherein we give you the data and you solve it. But again, chances in the past few years, right? If I have to take the past 10 years, this question or this concept has not come that much, which is why we'll quickly move on to the next one. But nonetheless, it's important, right? And if it comes, this is how, right? They will give it to you and you have to solve it. So now moving on to last activity from life processes, activity 6.8. And as we see, it is there in page number 109, right? So this is a very simple activity wherein we see that we have to take two small potted plants. Now this particular activity is based off on the concept of transportation in plants. Right, so it is based off this particular topic. Now what are they saying? We are going to take a potted plant. Okay, so we are going to take a potted plant in one, and on the other side we will take a pot which has only one stick. Okay, and then what are we going to do? Now they are more or less place a stick of the same height as the plant, right? Now cover the soil. So now we are going to cover the soil in both cases. So in both cases, I'm going to cover the soil with a plastic sheet. Yes. So I'm going to cover it with a plastic sheet. And then after that, I am going to cover both of this, right? So I'm going to cover both my plants with a plastic, my plant as well as my stick in a plastic sheet. So let me call this as A and let me call this as B. Now, do you observe? So what we're going to do is we're going to take these two pots and we're going to keep it in bright sunlight for some amount of time. Now, they're asking us if there is going to be any difference in these two cases. Are we going to observe any difference? Pause this video and you try to answer this yourself. But the answer is very simple, right? Now, we know that when of course in plants, a very important process takes place, which is what we understand as transpiration, right? So what is transpiration? Transpiration is nothing but the loss of excess water from the aerial parts of the plant. And of course, we know that excess water is lost in the form of water vapor and it goes throughout the aerial parts of the plant. Now, in this case, in the case of A, we see that there will be slight water droplets that will start appearing on the surface of the plastic sheet. And we see here that in this case, we see that there is no such observation. So we observe that transpiration is a process that we see in plants, while of course this is a control, right? This is kind of to tell you that it is seen mainly in plants and not just, in just a random stick. And why is transpiration necessary? Because in tall plants especially, it creates a suction pull that helps with the transportation, right? Especially absorption. We see that transport of water from the roots to the aerial parts, we see that the transpiration pull plays a very important role. So with this, students, we come to the end of today's video. Now, of course, as you know, we take your examination preparation very seriously on this channel and we've kick-started our SHIKHAR series on the same, which is why, as you see, every day between Monday to Friday, I would say between Monday to Sunday, you have classes on the channel that happens for you for across subjects, from science to maths to social science and English as well. And along with that, we give you a lot of extra information based on your board examination prep. So all you need to do is to stay subscribed to our channel. And if you found this video and you were here till the very end, let me know if you enjoyed this particular video, because BYJU'S 9th and 10th has always got you covered. So do not forget to hit the like button on this video. Do not forget to hit the subscribe button. I will see you all soon. But up until then, everybody, take care, lots of love, and bye-bye.